photosynthesis are water, carbon dioxide, and light. Plants obtain these reactants through the environment or other factors. The reactants are obtained, they enter the plants, which enables photosynthesis. Reactants are also obtained through the plant's roots. There we go. Plant anatomy and transport also plays a role in obtaining reactants. The leaves traps carbon dioxide and also traps light. The roots absorbs water and nutrients. The stem supports the structure and carry water and nutrients. In the leaves, CO2 enters the stomates and oxygen is released from the stomates. The food made through photosynthesis is transported throughout the plant. The food also goes through a tube which is called the phloem. The phloem distributes the food throughout the plant's body. The exymes or xylems, so the xylems transport minerals and water throughout the body also.
So for our hypothesis, we believe that the kale leaves would float or rise in more quantities at a more efficient rate than the spinach leaves because of the darker coloring in the kale leaves. The more darker and the more green means there are more chloroplasts and chlorophyll present in that plant.
So for the setup of our experiment, we used a glass beaker, syringe, a hole puncher, 0.2% bicarbonate solution, water, a petri dish, a lamp, and spinach and kale leaves. First, one group member poured 300 milliliters of 0.2% bicarbonate solution into our beaker, and we also added a drop of soap. Then we hole punched our kale leaves and made sure that we created 10 discs. Next, we removed the plunger from the syringe. So this is the plunger, the syringe. And then we added our 10 discs inside, and then we closed the syringe and added the plunger back. And then we added some of the 0.2% bicarbonate solution into our syringe, and we made sure there was no extra air in the syringe. The next step was to create a vacuum. So one group member had to put their thumb over the top opening of the syringe, and then they pulled back the syringe plunger and they pushed it back in. And then the next step this step might have been repeated 1 or 2 times to make sure that all the kale discs sank. And so after we got at least most of the discs to sink, we poured the leaf disc and the water back into the beaker. One group member made sure that we added water to the petri dish, and then we added that to the top of the beaker. And then we turned on the lamp and we placed the beaker under the lamp. And the next step was to record for 20 minutes and to see how many of the discs rose and floated. So our independent variable was the plant that we used, which was either the kale or the spinach. Our dependent variable was the rate of the reaction. And the constants between our experimental group and our control group was the amount of bicarbonate solution used, the light used, and the water temperature. So this is our data, and I have it in a chart, and I also have it graphed. And we found that the rate of the reaction with the kale was a little bit quicker. So they both started off about the same rate, but then the spinach leaves stopped having leaves floating to the top, while the kale continued to have all of the discs float to the top.
Alright, so the purpose of us creating a vacuum with the syringe was that we needed to create a vacuum to ensure that the gases of the cell inside the discs were removed. So as Victor said, in our different discs, the different cells have air pockets, and we had to make sure that all the air and gases were out of that. The leaf discs got carbon dioxide for photosynthesis from the CO2 in the water bicarbonate solution that the discs were added to. The discs were able to float due to the discs performing photosynthesis and turning added oxygen inside the cell, allowing it to become buoyant.
So for this lab, we basically just analyzed the rate of photosynthesis between a kale and a spinach leaf disc. For this experiment, we just altered an environmental factor where the leaf had no oxygen within itself. This caused the leaf to lose buoyancy, and because there was no oxygen in the reaction of photosynthesis, we didn't alter the part of the photosynthesis equation. And so With this equation, errors occurred, which was the temperature of the kale and spinach leaves, which could cause variation in our results. And also, the vacuum that we created could not have been as airtight as it should. And also, we had 3 trials, which one failed, which could be from our technique when we created the vacuum. And one of the reasons why it could fail is because the leaves were damaged by pulling on it too hard when making the vacuum. Our control group data on spinach had a slower rate of photosynthesis than our experiment group data on kale, which this basically is exactly what we predicted with our hypothesis. So to show, and the evidence was at 10 minutes, both data groups, which is kale and spinach, had 3 leaf discs floated to the top of the bicarbonate solution. But after 10 minutes, the rate of photosynthesis for the kale leaves increased and 7 discs floated to the top at 13 minutes. And for spinach leaves, there was no more discs floated to the top. At the end, the spinach had 4 leaf discs floated to the top, so 1 got added between the 10 and 20 minutes.
Continue, I will crop that up.
I blinked that.
Nope, I said wait.
Or the plants, and also
Or in those plant leaves.
So first, one group member went and took. Okay.
Okay, so for her,
10 and 20 minutes.
thank you.